Okay, this will be everything you need to know for Unit 2 of AP Biology. I'm going to start first with the structure of a cell membrane. There are a lot of parts to a cell membrane. There's the phospholipid bilayer itself, where the head is polar and the tail is nonpolar, and there's cholesterol inside of it. The cholesterol is what maintains the cell's flexibility. The more cholesterol, the more bendy a cell is. Then there's these glycoproteins and glycolipids attached to it. The point of these is they're like markers for the cell, so they know which cell is which, I guess. So a skin cell would have different glycoproteins and glycolipids than like a heart cell, than a bacteria cell, and that's how white blood cells can tell the difference between oh, this is a bad thing, or oh, this is a good thing. This is a channel protein. This is also an integral protein. That is because it goes in between the phospholipid bilayer. That makes it an integral protein. This would just be a surface protein because it's only at the surface. So this is water right over here. This is facilitated transport because it's going from high to low concentration and it's not requiring any energy, but there are some kinds of transport that do require energy. Next is surface area to volume ratio. It's pretty simple. You multiply the sides of each thing, so 1 times 1 plus 1 times 1 plus 1 times 1 for each of those 6 sides. You add them all together and you get 6. The volume is the length times width times height all together, and that gets you 1. And the surface area to volume ratio is just those two numbers, and in between them you put a colon like that. So that's 6.1 or 621. Then we have the surface area of this thing. It's the same thing. We go 3 times 4, and then there's 3 times 4 on the back side. Then 4 times 1 right here and right here. And then there's 3 times 1 right here, and some other stuff, and you'll eventually get 38 if you add all of those up together. The volume, on the other hand, is 3 times 4 times 1, right? Length times width times height, and that gets you 12. So the surface area to volume ratio would be 38 to 12, or 19 to 6. I did that by just dividing these two numbers. Next is hypertonic and hypotonic things. So if a cell has a lot of solute in it, it is hypertonic. And if it has not a lot, it is hypotonic. The way I think about it is hyper and hypothermia. So if you have hypothermia, you don't have a lot of heat. But if you have hyperthermia, that means you're overheating. So a cell that is hypertonic is, filled with solute like that. And just like someone who is experiencing hyperthermia, it takes in a lot of water, to fix itself, to get back into that homeostatic or dynamic equilibrium with its surroundings. And if something is hypotonic or is experiencing hypothermia, it will shrivel up because it's losing water to maintain a dynamic equilibrium. So just remember that if it's on the inside, and there's not a lot of solute, then it's hypotonic. But if there's a lot of solute, it's hypertonic. There's also isotonic. I didn't draw it here, but that's when these two are the same. There's still water going in and out, but it's always going to be around the same as the dynamic equilibrium point. Okay, so there are two kinds of feedback. A cell wants to maintain homeostasis, so if it's experiencing negative feedback, it is because something took it off of its set point, or, the point where it wants to be. So let's say you get really cold or whatever. You start shivering to warm yourself up, but the more you shiver, the more you warm up, the less you have to shiver. So this is the amount of the temperature of yourself after you experience something cold, I don't know, you go outside in the winter. Positive feedback, on the other hand, is when something starts and then it just keeps on going and going and going. It's like lighting a match in a forest and it's starting a forest fire, stuff like that. Or it's like a chain reaction of something happening over and over and over and over and over and over and over again. Or I guess just bigger and bigger and bigger. Climate change could be one of those things as well. Okay, next is anatomy of a nerve cell. This is a nerve cell right here. It starts with the soma, which is the cell body. Right in the middle here you have the nucleus, and right here you have sticking out, these are the dendrites. These are the little branches that absorb neurotransmitters, but we'll get to that. Connected to these things are this thing called an axon right here. there's two parts of an axon. There's the Schwann cell, which is covered in myelin sheathing, and the nodes of Ranvier. So once a signal or a neurotransmitter is received here and a signal goes out, it will shoot out here and it'll jump over the Schwann cell because of its myelin sheathing, and then it'll keep on going, and it'll jump over, and then it'll keep on going, and it'll jump over, and then it'll keep on going until it reaches the axon terminal. This is what happens after it reaches the axon terminal. So this is an axon terminal right here. Once it receives that signal, these little vesicles filled with neurotransmitters and these channel proteins will shoot out neurotransmitters through a synapse, right, or across the synapse. And these will be absorbed by the receptor proteins in the dendrite. And that after that happens, the neurotransmitter uptake pump gets these neurotransmitters back, and then the cycle continues, right? This is the voltage-gated CA2 channel. Yeah, I'm going to be honest, I don't really know what it does, but it's on here and it was on the test, so good luck with that. Okay, now I'm going to talk about diffusion and osmosis. Diffusion happens with small nonpolar molecules like O2 or CO2, and it just goes straight through the lipid bilayer like this. It goes from high to low, like I said. And there's also osmosis, which is a small polar molecule, but it can go through the cell membrane just like this. Or it can go through a thing called an aquaporin, which is just like a transport channel or a protein channel that just for water. And it will also go from high to low concentration, unless done through active transport, which would go from low to high, but I'm going to go into that later. Okay, this is the active transport that I was just talking about. Active transport is from low to high concentration, There are two main forms that I'm going to talk about today. There is exocytosis, where it just like kind of eats a thing and then brings it down forcefully into the cell, or through a thing called a proton pump, or just like a ATP-based pump, where ATP goes here, and then one of these ATP thingies, these little P's, or these phosphate groups, they break off, and it just becomes ADP, and that gives it the necessary energy to open up, let these proton pumps out, or let these specific protons here out of the cell to make a electrochemical gradient. An electrochemical gradient, there are two kinds of gradients. There's a concentration gradient, like right here. This is low, this is high, but there's also a electrochemical gradient where this is negative and this is positive because there's so much more protons here. So those will also be likely on your AP test. Okay, next I'm going to talk about water potential. This is the sign for water potential. The more negative water potential is, the more water will go into it. Like for example, right here, this water right here, or this solution has a positive water potential, and this cell has a negative water potential, like really, really negative. So water will at first go into this cell until they're both evened out and they both have the same water potential. So the one that has the more negative water potential, that's where it goes. So the equation for water potential is equal to pressure potential plus solute potential. Pressure potential is the pressure of the cell. If it's an open beaker, like this one right here, the pressure potential of this water is 0. And then the solute potential right here is negative I C RT. So those letters actually mean something. They're not just crazy speak. Negative I, or just I itself, is ionization energy. So if you put water, or if you put salt into water, it'll break off into NaCl and not just be NaCl. But glucose, if you put that into water, it's just still glucose. So this ionizes into two things, so you just have this number be 2, but if it was glucose or something covalent, then it'd just be 1. C, that's the concentration, that's the molarity of something. R is the pressure constant, something like that. And for the sake of biology specifically, it is 0.0831. The temperature, however, is temperature in Celsius plus 273, which is temperature in Kelvin. So that put into here plus the pressure potential will get you the water potential, and the more negative one is where the water will go. Okay, and that's basically the end of Unit 2 for AP Biology. That should be everything that you need for the test that you're going to be taking. Good luck.